Okay. Okay, so today we will do 2 lab work. The first work we will to do is the Kirby-Boer test, which is antibiotic resistance test. What I will do is I will do a demo. Now I want one bench of each of you can have one represent to do it. Then the other students just share the results. The reason is I don't have so many antibiotics, the disk there. But I want some of you practice. So if you're interested, let's do that. Okay? But I'll do a full demo, and Karen will take a picture, so I'll upload something on the Blackboard. So let's talk about that. So let's talk about antibiotic. So what is antibiotics? That is a metabolic. Well, we say metabolics coming from one microorganism. They used to kill or inhibit another microorganism. And here, usually the metabolics, which is metabolism products, coming from this microorganism. This is usually yeast and molds. So let's say penicillin, okay? And another microorganism, usually it's bacteria. So let's say penicillin for E. coli. So that's the story. Then Q, we call it cidal. We have a terminology called cidal. And inhibit, we call it static. So this is what we mentioned in the lecture. So we just review. Second, how we testing antibiotic resistance. We really want to test is MIC. That is called the minimal inhibitory concentration. And we have several methods to do that, which is mentioned in the lecture. And today we will do is called Kirby-Boer test. And I talk about this, I say this is a semi-quantitative test. So I'll tell you why it's called a semi-quantitative test, which means we know the antibiotics, we know the minimum inhibitory concentration, but we don't know the exact number, which means it's 8 milligram per 8 ppm, 6 ppm. We don't know about that definite answer. We know some type of the number. Now in the textbook, we know you have an E test, is that right? That is what you do is you have a strip there. Then this strip usually coming from, let's say 108, 64, 32, 16, 8. Then you grow bacteria there if you still remember. Then the first one, which is showing you a transparent zone, that is a transparent, which means here, around here, if it's transparent, then that number is the MIC. That's what you learn. And then we also could do a dilution tube. Dilution tubes, what we did is called a two-fold dilution tube. We can determine MIC. So those are the 3 things we talk about that. But today we will be focused on the Kirby-Boer test. So I'll show you how to do it and also do a demo. So how to do a Kirby-Boer test? Kirby-Boer test is usually it's a disk-based test. So the first one is you're going to grow a bacteria, a pig bacteria, from a plates or from a slants. It could be a colony from the plates or colony from the slants. Okay, we're going to go to this saline solution. So we added go there. This is usually 0.9% salt, so we also call it a saline solution. So we add it there, then we mix it. Now the very important thing, we need to know how many bacteria we add there, approximately. Because if we add different bacteria there, or we add a different amount, then the total concentration will be different. There is a standards. Usually we have this one, then we compare to so-called McFarland standard 0.5 turbidity cube. We are actually comparing is the turbidity. So let's say this is a standard McFarland cube. I don't have it. I just tell you we compare this to that. If the turbidity looks, use eyes to see, looks similar, this tube we can be used. And how many bacteria inside? This is equals approximately 10 to the 8 cells per ml. So which means CFU per ml. And that is a standard. Okay, this is the first thing they're gonna do. Second, how we're gonna do it? Then you have an agar. This is a very special agar, Muller-Helton agar. I call it MH agar, acronym. A Muller-Helton agar. It's a little bit special, not really like the neutral agar. Looks very similar. Then you use a cotton swab. This swab gonna touch the bacteria right there. Then you do a very gentle swab. Then you turn 90 degrees, do another swab. Okay, like that. Actually, it should be very intensive. Very, very intensive. I usually draw more. Okay. Then we put the bacteria there. Now we're gonna have to do is called dispenser. Dispenser adding disc onto agar surface. Okay. Now, what is the agar surface look? Well, what's a disc looks like? Every disc you will see looks like that. It's very tiny inside of this type of tube. Give you example. Let's say gentamicin. Usually write G means gentamicin. I'll just make sure its spelling is correct. So that's called gentamicin or gentamicin. And then you write a G, a 10. G is represent gentamicin. This 10 is a concentration, 10 microgram. Okay? So you use dispenser. We're gonna be press a button. I'll show you real quick. Then we add it there. Therefore, you will have so many. Let's say out of 3, okay? These discs there. Let's say we have a G10. That's gentamicin. The second one, what we're going to have is tetracycline. What I'm going to have is tetracycline. Tetracycline. That is 30. Okay. Then we have T30. 30 is a concentration. Then another one what I have is streptomycin. The concentration is also 10, so I have another one. Streptomycin. So that's why I have another one is S10. Okay, I put it there. Usually use forceps to press a little bit. We skip that one. Then we do 35 degrees Celsius, 24-hour incubation. Then what you're gonna find after incubation? You may see these transparent zones surrounding that. It may be very large, may be very little. It depends. Okay, if it's like that, now what are we going to do the next step? The first step is you're going to measure the so-called inhibitory zone, the diameter. Okay, so you're going to have to measure right here. Use ruler to measure the diameter. Then you have to compare the standard table. Compare standard table. Okay, I give you an example. Let's say tetracycline, which is T30 tetracycline. Okay, I compare that standard table. We have O, I, and S. This R means resistant. I means intermediate. This means resistant. I means intermediate. S means susceptible. Now I want to mention, for bacteria to antibiotics, we use the word is susceptible. We don't say it's sensitive. Why? Because lots of the people, when you do a muscle injection, muscle shot of penicillin, you have to do an allergy test. Some people are allergy sensitive or allergic to penicillin. You cannot use penicillin. At that moment, we say it's sensitive. So, if we say sensitive, which means it's human to antibiotics. For bacteria, we call susceptible. Okay? Now you compare the table. The table tells you the number right here. I remember most of the stuff, but I don't remember this number. Okay, this is 14. This is 15 to 18. And this is 19. So we're going to have to measure the diameter. Let's say the diameter here is 22 millimeter. That's your sample, okay? It's 22 millimeter. What that means? Which means this number is larger than 19. So we say this bacteria is susceptible to tetracycline. But how about if by any chance we get the diameter is only 12 millimeter? When you see the 12 ml here, this number is less than 14. We will say this bacteria is resistant to tetracycline 30 microgram. If this bacteria coming from this patient, then this patient cannot use tetracycline 30 because it's resistant. So here is the thing, although you see an antibiotic resistant zone here, seems like it's curing bacteria, but based on the national standards, it does not going to work very well for this specific patient. But how about if you're in the middle, let's say you get a 17, that means intermediate. It depends. It depends the history of this patient, whether they are resistant for the other antibiotics. If not, you cannot use it. If no, then you can use it. So that's what we do. So here, why we say it's a semi-quantitative, we know the number, but not very accuracy because we don't know. We know it's resistant, but what is the minimum inhibitory concentration? We don't know about the detail. Okay, so that's why I tell you, that's how it works. Now let's do a demo for you. So Karen here, I'll show you how to do it, and I want some of you could be coming here to do some of the tests. I will let you. Some of you, if you're interested, you can do it. So first of all, you can come here take a picture of this. This is a disc, very clear. Okay, this is a disc. We're gonna have to add in. So when we add in, we put it there. Put one there. Put another one there. This actually should be go down. I don't think this one works very well. So, okay. Then another one I usually put on the side. So you can take a picture right now for this one. We added 3 antibiotics tube into a dispenser. This dispenser usually is expensive. It's about $300 to $400, and this disc is also expensive. Now we have to do is a label. This is molar Halton agar. You take a picture. So molar Halton agar we usually label like MH agar, and I'm gonna add is E. coli. So I write EC, E. coli, and my name, and batch number 1, and date 11/12. Now I want you these label actually should be on the side or write a little bit small, because otherwise if you label too much, that zone will be difficult to see. Okay, so let me do that. Okay, yeah, that's after label. Now what we're gonna to do is we're gonna using a cotton swab. This is a cotton swab. It has been autoclaved. So we take one cotton swab out. So you can take a picture with we take a one cotton swab out. I put these on the side. So take one more picture. We put the cotton swab out. And then we will be touching the bacteria, let's say E. coli. Okay, so we do the E. coli. We're going to touch the E. coli right there. Now we put onto a saline solution. This is a saline solution. So let's say we added it. Okay, you can take a picture. I don't know how density it is, but ideally the amount of what you put, it should be following the McFarland standard, which means I should compare standard. I don't have it right now. Okay, so this one actually should be thrown away. I will just put on this side. Okay. Now I want you guys, if you want to do, you have to just make sure it's mixed very well. So we want to make sure you mix it. Okay. Now we're gonna add it onto the surface. How we add it? We take another cotton swab. Take another cotton swab. So we are using another cotton swab we add inside. Okay, take it out. Then what you have to do is you have to be add it like this, very, very gentle. So yeah, Karen can take a picture. Very, very gentle. This straightforward. Now they turn 90 degrees, do a second. But do not put this cotton swab back to the solutions, because otherwise you're carrying on more bacteria. Okay, so we can take a picture right here. And we are trying to cover the whole surface, so we do it like that. Okay, after doing this, then we're gonna put a dispenser on the top. So you can take a picture. The dispenser we'll put in there. Then hope it works. Press the button. You will see 3 antibiotic stick is in there. Can you see it? Then we cover it. You can take a picture of this one. Then we cover it, and then we do the incubation. And after 24 hours, you should see some of the inhibitory zone. You will see it next Thursday, hopefully. Okay, so I just put it right there. Okay, I don't have too many left. You're the first lab. I want everybody, some of you want to practice, you practice. But don't forget to label. Now, originally we should use faucets to press a little bit, but I skip that because I don't have the faucets. I have to get it from the old lab, so we just skip that. Okay, so this is the Kirby-Bois test for the first one. Now the second part of the fact sheet, what I write is antimicrobials. Using filter paper disc is the same thing, which means instead of you have antibiotics, we put formalin, put lactic acid or any of the antimicrobials there, you put it on the petri dish. It's the same thing. So we skip that. Okay, I usually do it, but I skip it. Okay, so this is the first part of the lab. So I just wipe this off. I'm going to do a second part. I will be maybe just put right here. I want to save some space. I don't have to write everything from the beginning. So I'm going to write here. Make sure, can you make sure that one, it works? If you can come in, just Karen, make sure the camera can cover that side. Can move a little bit. So this will be exercise 1. Now the second will be exercise 2. Exercise 2, everybody can practice. We're going to do a food analysis of bacteria. Okay, so we have is ground beef, and we also have is a hot dog. Now, why we have to do the food analysis of the bacteria? Now, you know, in the food industry, some of the foodborne pathogen could be there. It's not going to be in there. Those are the major commercial products. But possibly, for example, ground beef, you could have E. coli, O157H7. That is cause Shiga toxin 1 and 2 and cause H2S hemorrhagic uremic syndrome. Hot dog, we care about Listeria monocytogenes. Because that's a major risk, cause lots of outbreaks. And if you look at the hot dog, is a label. I'm not sure you really care about that. When you see any of the ingredients, you see sodium diacetate. Let's say you have a sodium diacetate, potassium lactate. These are, we call it antimicrobials, which should be used. Remember the 3 alternatives to control Listeria on the ready-to-eat meat we talk about on the slides? Those are antimicrobials, okay. Ground beef basically do not have any of the antimicrobials there, and this requirement is 0 tolerance. You should not have even 1 cell. And if you have more than 10 cells, that's not good. This is going to cause people to die. So remember the lecture, what I talk about is hamburger, the uncooked hamburger in a Jack in Box killing like 4 kids. That's a story is coming from E. coli 157H7. So I'm going to show you how we're going to do it, okay? The step is very simple. We're going to do the food preparation. So we're going to be preparing in food sample bag. Basically, we're going to add 100 ml nutrient broth in there. Then for different food products, we do, thank you, we'll do it differently. For you guys, we will be doing these 2. For next lab, we may be doing a tomato. Now, regarding the tomato, I just want to be very briefly. For tomato, the places where it's easy to have a bacteria in there for penetration is this stem skull. This stem skull area is very easy to have a bacteria penetrate inside. So the tomato industry, when they do the washing, they have a requirements of the 5 degrees Fahrenheit between the wash water, the temperature, and the tomato. Okay, so we just skip that. We're not going to do it today. So we add 100 ml nutrient broth. Then the preparing for hot dog and the ground beef, there is a difference. This is we're going to have to shake 30 seconds. This is we use a stomach. So we call it a stomach to break it down. And after that, we will have to do is 10 or 100-fold C area dilution. I actually want you just to do 100-fold C area dilution to prepare final dilution factor 10 to the 3, 10 to the 1, 10 to the 3, and 10 to the 5 on agar plates. And you can figure this one out how to do it. Remember, you're going to get those tubes, is that right? How to do it? This is the original sample bag. You have a hot dog there, let's say. If you add 0.1 ml onto the agar plates, that is 10 to the 1. Then if you add 0.1 ml onto 9.9 ml buffered peptone water, this go to the agar plates. That is the 0.1 go to the agar plates. That is 10 to the 3rd. So we need a pipettes. I'm gonna get it. Can you see that in the other lab? It should be there, otherwise I'll get from my lab. This comes here is 0.1, sorry, we don't need that one. Then 0.1 ml go to this one. And then 0.1 ml go to this place. That is 10 to the 5th. Okay. Then we all gonna do an incubation 35 degrees Celsius, 24 hours. Then you have to count the colony there. For you will be 1 week manual count colonies. Okay? So that's a procedure. Now I want to show you how we do that. So I'll give you some of the examples. This is a nutrient broth. And I will wear the gloves. This should be, I have a new one. Okay, let's do these. Do you have a scissor? Okay, that's good. It's not good. This has not been using for long.
Yeah, they're not great.
That's maybe 5 years. That's why if you see these gloves like this, has not been using maybe 5 years. So we have 2 different sample bags we'll be using. I mean, you maybe stick only one. Okay, I don't have that many bags. So this is how we're going to do the sampling.